Aloha, and welcome to the October installment of our monthly Slice of PI-CASC Seminar, hosted, yes, half a day, sorry, hosted by the Pacific Islands Climate Adaptation Science Center, or PI-CASC. For those of you joining us in person, please help yourself to pizza throughout if you haven't already. And as usual, in keeping with our theme, Slice of PI-CASC, we have some pie after the seminar. So stick around for that if you'd like. I'm Rachel Lentz. I'm with the communications team of PI-CASC, filling in for our usual MC, Deputy Director Brad Romine, who was not able to join us today. This Slice of PI-CASC seminar series provides a venue for sharing state-of-the-art climate adaptation research and science to management applications in Hawaii, the US affiliated Pacific Islands, and beyond. This presentation is being recorded, as you noted, and will be available on the PI-CASC website and YouTube channel in a couple of days. For those of you joining us online, please keep your microphones on mute during the presentation. We will have time for questions following the presentation, so please feel free to either leave your questions in the chat or wait till the end and we'll call on you. Today, we are excited to have as our speakers Dr. Laurie Raymundo and Ashley Castro, who are joining us online from the University of Guam Marine Laboratory. We're also joined by Dr. Romina King, our PI-CASC University Consortium lead at the University of Guam, who I will now pass things over to introduce our speakers. Romina, take it away.
Hey, so it is my honor and pleasure to introduce Dr. Laurie Raymundo and Ashley Castro, who will be presenting on challenges in conserving at-risk Acropora corals in a changing climate. Laurie Raymundo is a full professor of marine biology and the director of the UOG Marine Laboratory. Professor Raymundo is a coral ecologist who has spent most of her life in the Philippines. Woo! Love the Philippines. Okay, before moving to the University of Guam. I've known Laurie for almost 2 decades, and she has been a tireless leader in the Guam Coral Reef Initiative Local Action Strategy, Coral Bleaching and Disease. Professor Raymundo's father was an impassioned diver and underwater photographer back in the ancient days of strobe lights and film. She started snorkeling at the tender age of 6. Her career has been shaped by an early life immersed in both the incredible diversity of the coral triangle and the tragic human destruction of reefs. Her current research interests focus on reef rehabilitation and restoration practices as responses to coral loss from anthropogenic impacts, such as infectious disease and bleaching. Professor Raymundo has earned a Bachelor of Science in Zoology from the State University of New York Environmental Science and Forestry at Syracuse, as well as a Master's of Science in Water Resources Management from the State University of New York Environmental Science and Forestry at Syracuse. She has a PhD in Marine Ecology from Cornell University. So cold, Cornell. Professor Raymundo is joined by Ms. Ashley Castro. Ms. Castro is an avid diver, always jumping into opportunities to explore the ocean. She has a Bachelor's of Science in Biology from the University of Guam and is now pursuing her Master's of Science in Biology at the UOG Marine Laboratory, where she is studying trends and growth rates in Acropora's at-risk staghorn coral in contrasting ocean environments. Ashley works with nonprofit conservation organizations and is head of UOG Marine Lab outreach programs to reinforce community connections to the true value of what the ocean provides and preserve cultures and natural resources. Join me in welcoming Laurie and Ashley. Woo! Yay! Woo!
Is this where I start talking?
Hey, hi everyone.
Hi everyone. So am I sharing screen? How does this work? Can you see the screen? Am I sharing?
Not yet.
Okay. I think I need permission to do that from your end.
You shouldn't.
Oh,
I have not got anything
Oh,
requested.
okay. Let me see. How about that?
No?
Huh. Okay.
At the bottom of the screen, the green share button. You
Yep.
clicked on that, and then the slides
Here.
window. There we go.
There we
There
go.
we go. Okay.
Okay. No. Sorry. Okay. All right. Are we all set now?
Perfect. Go for
Okay.
it.
Okay. So today I'd like to talk to you about the efforts of many people at our lab that are doing a variety of really cool things regarding the attempts that we have initiated in preserving our staghorn corals. And I'd like to start out with a little bit of background about why we're doing this. So prior to 2013, Guam had not actually ever experienced really profound bleaching. We all know about the 1982-83 bleaching thing, which was the first big one. There was another one in 1998-99. And these were these big global events where lots of people were reporting lots of things dying. And Guam kind of escaped that. There was some scattered bleaching, but not a lot of mortality. And that very abruptly ended in 2013. For the period of 2013 to 2017, we had bleaching and extreme king tides and disease outbreaks every year for that 5-year period that left us breathless with the amount that we were losing in this very short period of time. And I have the dubious distinction of actually being quoted as the headline in the Washington Post's environmental section in 2016 because NOAA found out, Mark Aiken found out, and he said, hey, you got to interview Guam, because Guam's experiencing this. The rest of the world were just starting to gear up for bleaching in 2016, but we had started 3 years before that. And so, yeah, this is pretty much basically how we felt. We were going out every day and we were crying. I mean, I'd never done that before. That was an experience I'd never had. So in total, as we were doing these rapid assessments every year, by the time we got to 2017, we had lost an estimated of 36% of what we had had prior to 2013 in our staghorns alone. We were doing surveys for everything, but this was a quick and dirty kind of estimate of the staghorns because they're quite accessible. And I put the thing 27 football fields in there because, people can imagine this. And so when I talked about this before, that's basically what we're left. And Guam is, not big. We don't have that much coastal area. So as this was happening, those of us in the management, the management agencies in Guam, decided that it was simply, we have to do something about this. We can't just keep watching this happen because we don't know how long it's going to continue to happen, and we don't know how frequently it's going to happen in the future. So that was the context. And then, of course, I kind of was having trouble wrapping my brain around, well, where do we start? There wasn't any particular ecosystem, any zone that seemed to be more affected than not. We were seeing bleaching down to 60 feet when we were on surveys. There were multiple species. There didn't seem to be any species that wasn't affected. So my focus on Guam staghorns, was a calculated decision. First of all, because they amongst themselves form an essential fish habitat. It's not very many species. They are the major habitat structures, and they're very iconic. People know about them. So I figured we get some community buy-in and agency buy-in. They're also very accessible. So we were thinking about cost and about developing methods and being able to test them. We thought the accessibilities would facilitate our being able to develop best practices and experiment with methods without and minimize the cost and maximize the amount of time we could spend on these reefs. And then finally, we know that the Caribbean and the Western Atlantic have been doing this for quite some time. And rather than keep reinventing the wheels, there was an abundant literature that we could access and people that we could talk to about what their best practices were. So we thought, let's start with that, with the idea that we would be able to pass on these methods and that a lot of them could be used or modified for other groups of species. And that's, of course, as you'll hear from Ashley in a while, that is exactly what's happening. So basically what we had to do first was do a lot of surveys. So that's when I hired Ashley. She came highly recommended. I threw her in the water. There was absolutely no training. She just kind of started doing the work. And okay, you're going to do this for the next year, and it's going to be really boring. And she was a trooper and never wavered in her enthusiasm for doing transect after transect after transect. So we really wanted to get a really good data set for how much we had left. People would say, hey, I saw some staghorn, and we go check it out. And I think we got all, but I think there was 2 populations that we've found out about since then. So we've done a couple of other since, but we basically got pretty much everything. We were lucky because Guam is a discrete, fairly small island, and we could literally go to every single site. So the result of that was a monster tech report that we put out with pretty much everything that we had done, including initiate the nurseries that we initiated. And in a nutshell, this is what our surveys showed us. So this is what we found basically by 2021. So the size of the circle represents the approximate area. So some of the areas are as large as 75 hectares. So we did georeference everything. And there were a few populations that are pretty tiny, less than 1 hectare. And the color represents how much is left of living percent cover. So we found that there were 4 sites, one fairly significant site, that were totally gone. They were totally dead and just basically rubble and dead standing skeleton. And there were 4 species that were reduced to single populations out of the ones that we think we have. So originally we had 8 morpho species. 4 of them were now found in only a single site. So that kind of set the framework of what we could do and what we really needed to do. And as marine restorationists and coral reef restoration workers know, there is a bit of pushback about this as a science. There are detractors who think that reef restoration, coral restoration is a waste of time and a waste of money and we shouldn't do it. And I disagree. I keep thinking back to terrestrial restoration efforts, the fact that we have been restoring forests for several hundred years. And we know about wetlands, we know about river stabilizations, we know about grassland restoration. And corals are, basically really kind of more plants than animals. I mean, they're both, but they're very plant-like. So I keep thinking back to what we have learned from terrestrial systems when I try to apply things to coral restoration, but with the caveat that we know so much less about marine systems and even about coral organisms. Our background is incredibly inadequate, and we have to be responsive even as we're learning about these animals that we are, these plant animals that we are trying to restore. So we have the pressure on us to develop methods even as things are degrading, which is very often the way human beings, function anyways. And I keep thinking about things that my daughter, who's a plant terrestrial ecologist and a restoration person as well, and she'll say, well, Mom, what about your, community networks and how these species interact? And you can really plant with that kind of thing in mind. And I'm thinking, we don't even know that much about coral yet. We're so far behind. And yet I feel committed that this is the way that we have to do things. We have to keep soldiering on, and we have to take opportunities like this today to talk with other people that do it and share our results. So that is basically the background. And now I'm going to turn it over to Ashley to talk about sort of what we have done to date. So this is a cool shot. You want to take it over? Explain that shot?
Yeah, thank you, Laurie, and aloha, everyone. Please save me a slice of a pizza back there. So let's dive right into the restoration approaches we apply in Guam. As we all know, coral restoration is increasing with such enthusiasm around the world because as humans, we want to help assist in natural recovery and boost coral resilience in the face of climate change. So no matter where you are, whether you're a big island or a small island like us, you can't really avoid the consequences of climate change at this point. So here we talk about our restoration approaches and metrics in Guam. Thank you. So in Guam, we have 2 ocean nurseries established in contrasting turbidity, depth, flow, and management environments. In PD on the top right, this is a 5-meter depth. It is well flushed and it lies within a marine protected area. This site also provides sufficient water circulation and low sedimentation. Compared to our Maritzo nursery in the bottom right, we have a turbid environment where there's more silt exposure. The depth is at 10 meters. There's less water circulation, and this site is also heavily fished. Moving on to the next slide. Thank you. Both nurseries are established in areas that are sheltered and protected from typhoons and storm damages. My thesis study focuses on 8 Acropora species around Guam. And so this is where we harvest, as you can see on the top right, it's finger-sized fragments of each species from the surviving wild populations around Guam. And then we culture them on mid-water suspended coral trees. And then I take 10 of those and I monitor them for 12 months, and I get their total linear extension, which is the sum of all branches. And I collect data on that to see their growth trends. And so we're talking about 8 species, right? You're probably wondering what they are. In the next slide, I give you a visual. There we go. Of the beautiful 8 Acropora species that we have in our nurseries, we have Vani, Aspera, Vergata, Acuminata, Muricata, Austera, Pulchra, and Terrace. These are all beautiful species, and some of them you can tell that they look pretty different from each other. You have to be an expert at this. It took me a long time to actually become familiar with how they look physically, but I've been doing this for a long time, so I think I'm pretty good at it. In the next slide, we have a pie chart. This pie chart depicts our outplant progress between 2021 to the present day. And so this breaks down the percentage of species outplanted around the island currently. If you focus on the bigger slices, for instance, Aspera in red and Muricata in yellow, this is where we input monitoring for the species mortality and other observations to see how they fare in the outplanting process in terms of acclimating, surviving, and their resistance to bleaching and so on. If you see on the top, we have a peach colored slice. This is Pulchra, and they do really well in Guam, which is why it is such a small slice. We did not outplant as much because they're pretty much the favorites for the most part, and they give no issue. But if we were to outplant them, this specific species, it's to cross it with other species for experiments. For example, we have one grad student on Guam here focusing on white syndrome disease and the transmission rates in Pulchra. Moving on, let me take you on a tour of Guam and the sites we have outplanted in. I think Laurie has, okay, there. In red, we have the northern side of Guam. We have outplanted in. This is Tumon Bay. This is our tourist hub. These are where most of the hotels, the jet skis, and then also runoff posts as potential threats. But this reef is really helpful in finding resilient species of coral and is beautiful nonetheless. Also easily accessible, and this bay also experiences periodic algal blooms. The next green. This is the southern tip of Guam. This is Cocos Lagoon, where it was very salty, if you remember the photos. This is where depth variation plays a role in monitoring outplant success. And for these sites, we actually need boat access. So it's not easily accessible, but yeah. Next, moving on, we have, what's the next color? Yellow, PD, beautiful, beautiful site. So this is our most central point for our outplants, where there's good water flow, like I mentioned before. We monitor plots that are outplanted even right next to like a power plant, which is very interesting to see. So our goal is to outplant 4 acres of staghorn colonies, and we have fulfilled almost, I would say 80% of our goal, with about 20% remaining. And so you might be wondering, how do we outplant? In the next slide are our outplant methods. So on the top photo, you can see rope. We do rope structures and pavement, which involves cement mixing. And then in the bottom, we have C-frames. Can you see me? Can they see me? I'm going to do a visual with my hand. Okay, hopefully you can see me, but if you're not familiar with the C-frames on the bottom, I should have did a whole picture, but I didn't. So it's technically shaped like a C, and on the top it is secured with a plastic mesh. And on top of the plastic mesh, you can see in the photo that we secure colonies with zip ties onto that, and they're all equally spaced. Just to give you a better idea of how we, what methods we use to outplant. If you look on the pie chart on the right side, the rope outplants lie at 38%, pavement is at 30%, and our C-frames are at 31%. And so all 3 of these outplanting structures are also determined by depth. For instance, shallow outplants would need to be attached by cement rather than a C-frame, considering low tide events and reef flats, for example, Tuman Bay. So how are they doing? In the next slide, I will give you cool graphs. So this is discussing mortality. And as you can see on the left, mortality in Tuman, which was the tourist hub, like I mentioned before, was the highest compared to the other sites, mainly due to predation, bleaching, disease, stress, and other potential factors. The graph on the left side shows the mortality per site after a 1-year period of being outplanted. So keep in mind that these are also, the data here is also recovering from a Category 4 typhoon that hit us earlier this year, which really did impact our reef. So this can also be resulting in the death that you see in these charts. So on the green, on the right side, the green are the total outplants, and then in the red or pink, however you want to call that color, we see the survival success. So on the way right side of the graph, we see mortality that is just about equal to the outplanted colonies. And there are a lot of reasons for this death. It is impossible to pinpoint just one cause, especially for this site. But we can see that the other sites on the left side are essentially suitable for the other outplants. And so we learned so much about staghorn coral. It's amazing to discover what they like and what they don't like, but it is also really frustrating. So I'll hand it over to Laurie again, and she will show you what we have learned about staghorn as a model restoration species and what new knowledge we just discovered and is surfacing from the research that is being done at the University of Guam.
Thank you, Ashley. Yeah, so when we started doing this, I was working with a variety of our local resource agencies. We were all talking a lot about what we wanted to do. But because restoration is something that is currently kind of big in the funding agencies and there is funding available, a lot of my colleagues who are a lot smarter than me at the lab started saying, hey, you know, why don't we try to this and let's see what we can learn about this, that kind of thing. So the next few slides are just talking about all the different things that my colleagues have been involved with and what they're starting to tease out. So the first thing is, you know, we have morphospecies definitions. These are based on these standard classic morphological characteristics and what we think we have. However, none of this has actually been verified. This is all our best knowledge of work done by Dave Burdick and Dick Randall, and talking with the Aussies and various other people who are working with staghorns to try to figure out, are these good names? Are these the solid names? So we've originally started out with 8 morphospecies, not really knowing whether these are true names. So we don't really know if these names are correct. We don't even know if some of these may be endemic. We didn't have any idea how clonal these large thickets were. Staghorns, as you know, tend to grow in very large thickets. And were these all clonal? Was there reproduction, sexual reproduction going on? We didn't know how these populations were linked. So the population geneticists wanted to address some of these questions. For now, we're basically all using CF for all of our species. We have deposited specimens in the Phoenix project. I'm not sure if you're aware of this, but there's a group of people all over the world who are trying to resolve some of these issues of taxonomy using molecular tools for at least Acropora, which is the biggest group and the most messiest group. So it's a daunting task. So the jury's still out about what the names are. These 4 species are the ones that are, let's see it, that is Acuminata, Aspera, Austera, and Vani. So these 4 species are basically the ones that are found only in single locations. There are no other wild populations that we've been able to discover where they exist. So these are all considered the most at risk. We don't know if they're endemic and they're only in one location, and we don't even know if they're found any more northern than Guam. So we're not sure if they're in the upper islands, but they don't seem to be. They haven't been recorded by people in Saipan that are working on this. So the Kambosh lab, the Island Evolution Lab, has started to tease out some of these questions. Master's student Darian Rios looked at Acropora pulchra, which, as Ashley indicated, is the most common. That's the one that's doing the best. It's found in multiple sites all along the western coast, and there's one population on the eastern coast. And she took the first look at population structure within this species. And really coolly, she found, so she also looked at Saipan. They were able to get Saipan, specimens from Saipan, which is that separate sort of salmon-colored blob on the NDS graph. And that's distinct. So we know that that's genetically distinct. And we see that the green, which is the most southern, and the pink and blue are the northern, and then there's this yellow circle in the middle. And interestingly, sort of the northern populations are distinct from the southern, and then there is some indication that the population in the middle kind of links the two. So that's cool. So our southern and northern are distinct, and there's a population where there's apparently some gene flow that links the two genetically. So there is some connectivity, and that was interesting to see that mix. And the other thing that she found was that there was distinct clonality within these populations. So these massively big thickets are actually predominantly clonal, which says something about what we need to do in terms of assisted sexual propagation. Now, Hector Torrado, who's a postdoc in David Kambosch's lab, and the rest of their lab has been looking more closely with clonality in these populations. And this one in particular is Acropora aspera, shown in this photo here, which, if you remember from the previous slide, is only found in our southernmost population of Cocos Lagoon. So there's one population left, and yes, it is highly clonal. So Acropora aspera is an example. That's the one that we've been able to look at the most. Very clonal and very alone, the only population left. So this again provides us with some ideas of, okay, how do we manage this? Some concern and some ideas. So master student Michaela Ruder looked at trying to figure out how these 8 morphospecies were connected phylogenetically. So that's still work that she's furiously finishing. She is now in a PhD program, but she's finishing off her thesis first. She's a busy lady, and she's done some great work. And amazingly, her early information, the tree, which I won't subject you to because you won't be able to read it. It's tiny font, and they're messy. It's a big tree. We're not going to do that. But what she did find from the phylogenetic work is that 4 of our species seem to be linked. So Acropora and Pteris apparently are the same species, phylogenetically, molecularly, and Virgata and Muricata are apparently the same species. Morphologically, they don't appear to be. What she's found, so basically, she had few samples, so we need to sample a bit more. But what her analysis has shown is Muricata, this one with several different colors, have distinct population structure. Between the populations, there's distinct genetic differences. So this is good. That means that we may be able to put them together and increase diversity by sexual propagation. But Acuminata, aspera, and austera are not in good shape. They are single clones, and they are also single populations left. So this is kind of a sad warning sign for these 3 species. Virgata is the other one. No, Vani is the other one that she wasn't able to analyze in this particular data set. So it's not looking good for these three populations that are found in a single place and are highly clonal. So it was really good to figure out some of those, tease those kinds of things out, and it's been very helpful. So what about sexual reproduction? Nothing was known about when these guys spawn. So the first thing we had to do is figure that out when they do spawn. And this was early work done by Val Lepasek, another master's student, who looked at 5 species. Well, actually, it was 4. Turned out to probably be 4. And she did it primarily from histology. So she sampled a whole heck of a lot of gonads and looked at when they were ripening, when they were maturing, when they were no longer present, and figured out spawning timings. For 3 of them, Pulchra, Muricata, and Acuminata spawn in April and May. And Aspera, sadly, spawns in September and October. And so Aspera not only could not potentially hybridize, but it's also, it spawns at a very different time. So since Val's histology work, we have been going out in April and May, and we figured out after 2017 that even though most of the rest of our species spawn in 7 to 10 days, 6 to 9 days after full moon in the summers, we started doing that in April and May, and no, it ended up that they are very politely spawn on full moon. And possibly the day after, if full moon tends to be the next day in the middle of the day, you know, it's like when is the moon biggest. So it's full moon plus 1 day. We usually go out both of those days. And we've been going out as much as the weather will allow for April and May. We've been able to figure out now additional spawning in our nursery, which was really exciting, and in one of our outplanted populations. Pulchra, acuminata, virgata, austera, and vani all have spawned within our nurseries. We have not been able to catch myricata and not terrace. So there's kind of holes in this. I mean, we're figuring it out. We're going to keep going out at night. It's around 8:30 in the evening, quite polite. But we're not getting consistent spawning in both months or in either month consistently. So there's still a lot of holes in this. Yes, they are probably certainly impacted by bleaching, which happens the year before and could impact the following year's spawning. And certainly from our major typhoon in 2023, that definitely set everybody back. So we've still got a lot of holes to fill in regarding when they actually go. We know the timing, but the months are still, yeah, it's kind of a crapshoot. We're not really, there's not a consistent pattern. So Mikay evidently thought that she didn't have enough to do with the sea fish she was doing. So she did a sea bass short-term heat assay. She was interested in how these 8 species at that point fared in terms of heat resistance. And we had Mike Sweet and Chris Vulstra come out and teach us sea bass. We did that with a subset of these populations. And she said, why don't I just do all of the species and work with the ones we have in the nursery? So she did this, and it was very interesting because what she found is distinct differences in heat tolerance, which has guided us in regarding where we decide to outplant these. We can look at this, look at her data and figure out, okay, well, this is good, and that wouldn't be good at this site. So, but interestingly, I know you probably can't read the script on the bottom, but basically, so these are the 2 species. This is Pulchra and Terrace that fall out kind of in the middle in terms of how resistant they are to heat. They are supposed to be the same species, but whether this is a zooxanthellae difference, we're not sure. We haven't tested for what clades they have in the nursery, but so that these are close, but they are a little different, despite being the same species. But a more drastic difference in heat resistance is virgata versus myricata. Virgata is basically very, very susceptible. Myricata is tougher regarding heat resistance. And yet these are supposed to be the same species. So despite what the molecular evidence tells us, there are population differences in terms of how they're responding to heat. And again, we need to, learn a little bit more, but that's starting it. So this was getting back to more why we saw that 100% mortality in Tumon. This was work that we did with, this was Aspera. Okay, so we worked in Aspera first. We had original records that Acropora aspera used to be in Tumon. And since we've figured out that wasn't the case, but we thought that they would do well in Tumon based on the conditions of the wild population. So we planted them out. We were, okay, this is our first attempt to plant into Tumon. It's a marine preserve. It's a very iconic site. Let's do some outplanting. They did well in the beginning. When they were planted in February, we wanted them several months to acclimate before the summer bleaching season. And then May, we noticed some very distinct white syndrome. And a couple weeks later, we started seeing them bleach, and then they were completely bleached. And by August, everything was dead, the entire population. Now, we didn't know at the time what we know now, that this is a clonal population. So even though we were hoping to see some differential responses to disease and bleaching, basically we saw no differences because they were clonal and everything was gone. And it was this combination of white syndrome, and bleaching, but there were water quality events. This was timed during, we had a red tide bloom twice, and the water gets very flat and calm. So we realized, okay, this is a combination of circumstances we should not try, even though we hope that restoration can work in sometimes less than optimal conditions, because a lot of our reefs are less than optimal at this point. This was a case where we said, okay, can't do this again. And so we saw absolutely no differences in responses. But the interesting thing is that we did have, with my colleague Eloise Rosé, said, well, let's look at the microbiome, because that's what I do and I'm really interested in it. And what we found is that when she looked at the wild population and what was in the nursery and what was happening in the diseased, she did see signs of dysbiosis. And she saw dysbiosis early on. Well, she saw increased diversity of the microbiome community early on and in the nursery, and a lack of some of the bacteria that we associate with healthy corals. And we hypothesize that this might have had something to do with how susceptible they were to disease. So that kind of stimulated her to do a little bit more with this. And we did try to outplant again in Tumon 2 other times in different locations, different species, and none of them have thrived. So Tumon is problematic. Amazingly, there is a lot of healthy coral growing there, but it's not an easy place to transplant into. So we're not going to work with that site until we get a better handle on what's happening and why, and how we can avoid that. So continuing on with the microbiome story, there's a group of us who are looking at another species, so Vergata, which we know now is technically Muricata, and looking to see if how microbiomes change with movement into the nursery, and then can we do something to facilitate healthy microbiomes in the process of outplanting and reduce the amount of stress and the responses that we kind of collectively refer to as transplantation stress, sort of a failure to grow, maybe some bleaching, some tissue loss, and these kinds of standard responses that we see in outplanted corals. Might knowing more about the microbiome and how to keep it healthy improve our success rate in establishing new populations. So the first thing that was done was basically to look at the wild populations and then move wild population fragments into the nursery and look at the differences between wild microbiomes and what happens when you move these colonies or fragments into the nursery after a period of months. And interestingly, this is what happened. Such a cool graph. I had to really study this to understand it, but basically in a nutshell, the source colonies, Opera and Coco, so these are our 2 wild populations, were distinctly different, but fairly homogeneous and dominated by Endozoicomonas, which we are learning is a bacterium which is associated with healthy corals. Now, when you move them into the nursery, all kinds of crazy stuff happens. You lose a lot of the Endozoicomonas, and you start seeing a lot more diversity in the bacterial that make up the microbiome. So all kinds of shifts and a loss of the healthy associated bacteria. So moving them into the nursery, even though they grow well, as Ashley's findings have noted, stay tuned for her thesis, they're not keeping the same microbiome. And it might have something to do with the fact that they're hanging in space, they're not firmly fixed onto the substrate and associated with substrate as a bacteria source. this is kind of what we're hypothesizing, and this is kind of what we're thinking might be the next step in this experiment. So this could explain why some of our corals fail to thrive when they're outplanted to a new location. So it's very exciting. Okay, so in summary, what all of these studies, a lot of them done by master's students, have shown, population genetics have shown us we've identified our species boundaries. We seem to have 6 rather than 8 species. We have high clonality in these very, very large populations, with the exception of one population, which is in a fairly water quality stressed area. West Agana, which is right next to a sewage plant, has a lot of genetic structure and is probably very successful in spawning. And we have identified 4 species of particular risk of extirpation on Guam, but we still don't know what their names are. So we're hoping that we find that out at some point, because that may determine whether or not they're even different than what we think they are and possibly endemic. So that leads us to, you know, how can we manage these species? So reproductive biology. So we have a bunch of probably clonal, highly clonal, massive populations that are separated from each other by kilometers. So the possibility of them, even when they spawn, of meeting in the night someone that they could fertilize is quite small. So low probability of fertilization success without some intervention there. We know they spawn, and when they spawn, they really go, but they may not even go on the same night or the same month. And yeah, so successful fertilization it seems to be fairly low. And we do seem to have differential resistance to bleaching. We have a student right now that is working up data from different populations of pulchra and see what their differential susceptibility is to white syndrome. And we know that there's some bleaching differences. And so we're now figuring out how to work that into our planning. And we know it appears that the microbiome has a major impact on this. Okay, over to Ashley to talk about what else this has spawned. Haha, unintended.
Yeah, all right. Haha, funny. So we've had the pleasure of hosting our first Pacific coral restoration workshop last year. Partners from Saipan, Hawaii, and Florida came to exchange restoration practices, and we all learned from each other. It was a really, really fun time. You can see them in the background, the whole picture. In our rope nursery, we all outplanted those together. That's some of the folks up there on the top right from NPS here on Guam. If you guys ever decide to come to Guam, please let us know. We're only 30 some miles long and 12 miles wide, so we can do everything in a single day. Next slide. And so we also have Guam Coral Reef Initiative just install their nursery right next to ours in PD. And they populated their trees with branching and boulder species earlier this year, and they are just doing amazing work. They are such a great team, and I look forward to seeing what else they have in store. Additionally to that, in the next slide, we have National Park Services. They also intend to populate their nursery they installed this year after the bleaching season passes. And so right now they're utilizing these months to just survey the island and to identify resilient colonies before collecting their fragments for their coral stock. So really cool things happening right now. In the next slide, we have, how do I say this? The Pacific region is coming together to work even more closely in hopes to push forward in coral restoration with more collaborations and more fun meetings. And I myself and the partners here on the screen will be attending Reef Futures in Mexico in December to meet and, you know, feel inspiration between each other and discuss strategic plans and techniques. to improve our outcome for the future in the world of coral restoration. And so I'm very excited to see what's to come as we approach a new year with these partnerships. And I know we are running short on time. We want to leave some time for questions, but I'll go ahead and hand it over to Laurie to finish it off.
Thank you. Well, so obviously there's a great deal of work still to be done. What we have started with the knowledge that we have gleaned from all of this really cool work that's been going on. One thing that we've decided is that we really have to facilitate sexual propagation. We have to bring these remaining, however much genetic diversity we can glean from what we've got left. We've got to facilitate spawning. We've got to facilitate fertilization success. So one of the areas in our PD nursery, so most of you don't know about this particular area, but it's got a number of sinkholes. So it's this wide, flat reef flat within an area that contains an area where these sand-covered sinkholes have just formed over, within the karst topography of that section of the island. And so these are deeper areas that are really, really good for coral. And so we've dedicated one to just planting as much of our genetic diversity as we can to kind of let them do their own thing when they do start spawning. We hope to capture that, but meanwhile, we're sort of passively sitting back and saying, here, you guys can get together as you normally do in these areas. And I mean, it does spark a coral dating service concept. So this is basically what it is in my mind anyways, strangely. But we are also going to be doing more as we learn more about when they spawn. We're going to try to do more ex situ culture. Our plans were thwarted last year because of the typhoon. This year, we were getting ready for spawning, sea-core spawning in July, so we didn't try to do it in the lab. Next year, we hope to just bring colonies in and see if we can do this in the lab. Right now, we are basically have really hot water. I don't know how Hawaii is doing, but Guam is bleaching, and we had a little bit of a reprieve, and now we're back into alert 2, and we've lost most of what we've settled in this year's spawning, so that's very disheartening. But the staghorns don't spawn during the bleaching season, so we can hope for more success in that regard. Some of the other things that we want to do, we're looking into cryopreservation. And we have to work with other people to do that. I want to initiate some conversation with Saipan about possibly crossing the species that we do have and seeing if we can introduce some more genetic diversity that way. Still working on getting names for our species. We're talking with Aquarium of the Pacific with some possibly sending over some fragments to them to let them start some ex situ displays and keeping some of this stock preserved in a really good facility. And thinking about incorporating a transitional stage to our outplanting between the nursery and the outplanting stage to try to manipulate that microbiome situation. So that is probably going to be a next stage in what we try to do. Okay, so yeah, these are all our people on the team that have helped us over the years, that have been part of this team. And we are so thankful for everybody. And these are our funders and our supporters and our colleagues. So yeah, we'll take questions now.
All right. Let's thank them.
Thank you.
So thank you very much. Now is the time for questions from our audience. But actually, since we are getting close to the hour, let me just make this announcement first, and then we'll go to questions. So next month, the first Tuesday of the month is Election Day. So please go and vote. But it means that we're going to have our November slice of PI-CASC on the second Tuesday, November 12th at noon, Hawaii time, which will be bringing you a different look at some Hawaii coral restoration work by Dr. Ford Drury and Kira Hughes, who will be joining us from the University of Hawaii Coral Resilience Lab and tell us about the ecological and sociocultural response to transplanting corals to enhance reef resilience. Those online, when you go off Zoom, will put up a survey. Please fill it out. Your suggestions have actually proved very useful to us this fall. We've actually done some talks based on those. And for those of you in the room, stick around for pie. But let us go to questions. Is there anybody online or in the room who has questions?
I think
Here's a
Mark has,
question.
There is
Yeah.
one from Mark. I'll just read it for you. How much variation is there in linear growth rates among morphospecies?
Ooh, you have to wait for my thesis proposal. No, I'm just kidding. So it actually differs greatly between each species. And yes, Laurie did mention that some species that we think are different might be the same, which is a complete headache for me. But I will present what I have known. But we can see, especially in the different sites, they definitely grow differently because of their environment. So let's say I have Muricata in the Cocos nursery and then Muricata in the PD nursery. Sometimes their growth rates are very different, where if they're spoiled in a little marine protected area, they just won't grow as fast as a Cocos one would because of there's a lot of competition for light, and so they extend more like branches. And so if that answers your question in a general way, that's pretty much my research. But stay tuned for my thesis proposal. Thank you for your question.
Anybody in the room? If not, we'll go back to the other one that's online. No? Okay. There was another one online from, I'm going to mess your name up, Dacia. Have you detected any wild recruitment or natural recovery in any of the coral beds at your survey monitoring sites?
so yeah, I'll take that one. Yes. So we have seen, they can resheet over dead skeleton, and we have seen that to a certain extent. Unfortunately, a lot of my monitoring indicates that a couple of these sites have continued to degrade. So mortality actually has continued after the 2017 event, and we think that some of that might have been disease, some of it could have been predation or nuisance algae coming in, because we have had all of these things happening. But some of them do resheet, so they can actually overgrow a skeleton that has been completely bare. Recruitment, oh my goodness, every once in a while I look for recruits. There are several sites that I look at twice a year in a long-term monitoring, and we have occasionally seen recruits, which is super exciting. Guam has traditionally been recruitment limited, and we have noticed this in a variety of different surveys by different people over the years. We don't have high recruitment. So when I see an Acropora recruit, it's really, really exciting, but it's quite low. So some sites are recovering naturally, but others have continued to decline. And I think that that's a pattern that we see in a lot of places.
Thank you. All right, well, we have hit the hour, so I think I'm going to have to cut it off there. But thank you for coming to us from Guam. We appreciate you getting up in the morning and telling us all this fascinating information.
Thank
so
you.
thanks very much. Should we thank them once again?
Thank you.
And we hope to see you all back next month. Take care. Aloha.